Chapter 7 is on the growth of bacteria as well as the culturing of bacteria. So just like you might have your ideal conditions for studying, so you've got your snacks, your lighting, bacteria, they are going to have their ideal conditions for growing. In lab, of course, we're going to be interested in growing bacteria. In some cases, we may want to quantify the bacteria that we have, and so we're going to have to employ some different methods. So with this chapter, we'll talk about some of the different growing conditions, the growth curve, as well as some lab techniques that we will actually be doing in lab. So a little bit on the division of bacteria, and I know that I've mentioned this before with the shape and arrangement. The arrangement of the bacteria, so whether it's in a diplo arrangement or Staphylo arrangement, has to do with those cells being stuck together with incomplete separation. So when they go through that cell division, prokaryotic cells divide by binary fission. And before the cell divides, it has to replicate its DNA. So this is important for any cell because if it has half the amount of DNA when it divides, it's not going to be able to survive. So first it's going to undergo DNA replication and then it's going to divide. This is a very simple, a very quick process as well, which is called binary fission. So if you look at this picture, the two daughter cells are identical to that parent cell. Now, different arrangements happen when those cells get stuck together. So when that binary fission is incomplete and the cytoplasm gets stuck. So that's when we would actually see those cells sticking together. Now, as we talked about before, we would see more arrangements with cocci because they're circles than we would with the bacilli because they divide end to end. So they would only get stuck end to end. So that's why with our bacilli, we would only see diplo and strepto. We would never see a staphylo arrangement with our bacilli. And here are some examples again. So we've already talked about the linked bacilli. We've also talked about the cocci. So the reason that we get these different arrangements and how those cells get stuck together is that they get stuck during that cell division. So they're not completely divided into those two cells. Of course, we know that some of them like to divide and they will separate very well. And so we tend to see those in a mono arrangement because they can separate very well. Here you can see that we've got some tetrads, we've got staph, and then we've also got some strepto as well. Yeast, which are microorganisms, they are very small. They are eukaryotic cells. They will mainly divide by way of budding. Some bacterial cells will actually divide by this form. And if you take a look at this micrograph, you can see some yeast. And you can see like the parent, the mother cell, and then you can see this little tiny cell which is budding off of that mother cell. This is cell division by way of budding. We would see this with yeast, with some prokaryotic cells, but mostly our prokaryotic cells are going to be dividing by way of binary fission. So a little bit on the growth of bacteria. If we were to look at bacteria in a culture and we were to quantify it and we were measuring it at different phases, we would be able to quantify that bacteria. We would notice some differences in their numbers. If we're talking about like leftovers that we might have for dinner, there's bacteria in there. Yes, our food is not sterile. There would actually be bacteria in there. They would actually be growing. Mostly we talk about quantifying bacteria in a culture in lab. But of course, we can have bacteria growing in our environment. We can also have it growing in our food. So these are the different phases or different stages of the bacterial growth curve. So the first phase we call lag phase. So this is when the organisms are kind of waking up, they're getting used to their environment. They're not really increasing in big numbers yet. They're just kind of getting used to the environment and becoming metabolically active. This phase really can depend on the type of bacteria. It can also depend on the conditions as well. So it could be very short or it could actually be very long. Log phase, this is when growth really takes off. And so organisms are going to be growing at a logarithmic or exponential rate. This is actually where we could calculate the generation time. And the generation time is the time that it takes for organisms to double their population. Now, the average for this is maybe an hour or two, but it can be very quick and can be 20 minutes. Some can be very slow growers, like tuberculosis, can be more like 20 hours. So what this means is that we could have a culture of bacteria, and within a matter of an hour or two, they can actually double their population. This is something that I think of when I'm going to a picnic and it's really warm outside and we've got potato salad sitting outside. I'm automatically calculating that generation time. Like, this potato salad has been out for quite a while. I think maybe it's time that we stick it back in the fridge. I also think about this with buffets. I'm probably not the best person to be taking to picnics and buffets because these are the types of things that I'm thinking about. Now, when these bacteria are growing and when they're in this logarithmic phase, it's not like they're all dividing at once. So it's not like they're all getting together, like rowing a boat and saying, okay, 1, 2, 3, divide, 1, 2, 3, divide. This is what we would call synchronous growth. This means that they're all dividing at the same time. This does not happen in nature. We can induce this in lab with drugs and chemicals, but in nature, we would see non-synchronous growth. So one over on one side would be dividing, and then one next to it would be dividing. So they're dividing at different rates. This is what we would call non-synchronous. Now, eventually, they're going to start to run out of resources. So water is going to start to become scarce. Oxygen, waste products are going to start to build up. And pretty soon we're going to reach this plateau. This is the stationary phase. And this is when the number of cells that are being reproduced equals the amount of cells that are dying off. So it reaches sort of this plateau. This is that stationary phase. Now, I know that we talked about our endospore forming bacteria. Remember, endospores are a protective feature. So if cells sense that, okay, nutrients are scarce, we don't have as much water, that's probably going to happen in stationary phase. And if they are spore formers, some of them are actually going to start to go into that spore state. We would actually see even more endospores form in decline phase. Depending on the textbook, sometimes you'll see death phase opposed to decline phase. Either one of those, they both begin with D. This is when cells are dying off at a logarithmic rate. Waste products are building up. They don't have nutrients. They also don't have water. So now they're dying off at a logarithmic rate. If they are endospore formers, they are really going to be making a lot of endospores. So they're just going to kind of go into that dormant state. If we were to take a sample and we were to look at cells in decline phase versus log phase, they would actually look much different. So when cells are kind of deprived of the things that they need, they're going to look a little involution. So they would look a little different than they would as those healthy cells in log phase. So I'm a visual person, so this is what I always think of when I think of growth curves. So here is this kind of like a bell curve. So first you can see that lag phase. So it depends on the conditions. It depends on the cell. Lag phase could be very quick. It could also take a little while. So lag phase, the bacteria is just getting used to the conditions, starting to divide, but we really haven't taken off at a logarithmic rate. Log phase, you can see where it's really taking off. And this is where they're increasing in numbers exponentially. This is also where we where we could calculate that generation time. So when I was in micro, we actually had to calculate generation time based on bacterial growth curves. You don't have to worry. I'm not going to make you do that. But if we wanted to find out the generation time, this is how we would do it. We would look at the log phase. We would look at the number of cells versus time, and then we could actually figure that out. If you notice at the top where it sort of straightens out and we reach that plateau, that's stationary phase. So because conditions are starting to decline, now we've got some cells that are reproducing, but some are dying off. So they kind of equal those numbers. So that's where we kind of reach that plateau. If they're endospore formers, some of them are probably going to start to go into that dormant state. And then finally, we've got decline or death phase. This is when they're dying off at a logarithmic rate. If they're endospore formers, we're going to be seeing a lot more. So if we were to take a sample of a spore-forming bacteria in a growth curve, we would probably see some spores during stationary phase, but we would certainly see a lot more in the decline or death phase. All right, so how can we actually quantify bacteria? So we're growing them in lab and we're interested in making those count. So how as scientists can we actually do this? Quantifying bacteria can be pretty tricky because we don't know if we have a lot of bacteria or if we have very few bacteria. One of the methods that scientists use, microbiologists, and we will actually be doing this in an upcoming lab, this is a serial dilution. So when we quantify bacteria, we need to get them to countable levels. I usually talk about this sweet spot of 25 to 250 CFUs. Now, a CFU is a colony forming unit. So if we are counting a sample, we have absolutely no idea how much bacteria we have. So we are going to have to hedge our bets, and we're going to have to do a serial dilution so that we get a range of bacteria so that we can capture them in quantifiable numbers if there's very few bacteria and if there's quite a bit of bacteria. So if you take a look at this picture, this is where we're actually doing a serial dilution. We're taking from one sample and putting it into sterile water and diluting the sample, so diluting the number of colonies. So if you take a look at the original culture, if we were to do a count on that original culture, there's 50,000 bacteria per mil. That's the approximate number. That is way too many to count. That's way outside of our sweet spot of 25 to 250 CFUs. So we would do a serial dilution. We would take 1 mil from the original culture, put it in 9 mils of water. This would give us a 1 to 10 dilution. This theoretically could bring us down to 5,000 bacteria per mil. That is still way too many to count. So we're going to continue, and then we would take 1 mil from our 1 to 10 dilution, add it to 9 mils of water. This would give us a 1 to 100 dilution. Now we're in a little more quantifiable range of 500 bacteria, but that still is too many. So we would continue on with our dilution. If you take a look at our 1 to 1,000 dilution, that's going to hit our sweet spot. So if we were to take a mil of that and we were actually to plate it out so that we could count it, that would be our sweet spot. If we continued and if we plated out the 1 to 10,000 dilution, we would only see 5 bacteria. That's what we would call too few to count. It's not statistically reliable. So you can see based on this example, we're getting a range of bacteria. When we're quantifying bacteria, because we have no idea how much bacteria we have in our sample, we need to do a serial dilution in the hopes that one of those will actually hit that sweet spot of 25 to 250 CFUs. So how could we actually plate them out and count them? So in our lab, we use our agar plates, and in those agar plates, we have nutrients. So we use media that has nutrients that the bacteria will like. And it also has that thickening agent, that agar, so that we could actually see the colonies growing on the agar. There's a couple of different methods. One method is a spread plate method. So that we've made our agar plates, we would take a small aliquot sample from our dilution, and then we would plate it out. We'd spread it out over the plate, and then we could actually grow it and count those colonies. Probably a better method, and the method that we will be using in lab, is the pour plate method. This is when we would take a sterile plate, add an aliquot of our sample, and then we would add our liquefied media and allow that media to solidify. This is a better method because, first of all, if we added like a mill on top of an agar plate, it might be kind of messy. The second thing is that this is going to allow us to capture the aerobic and anaerobic bacteria. Some that like the air, they're going to be growing towards the surface. Some that like it a little more anaerobic, that will shy away from the air, they will grow inside the media. So it's just a better way of capturing all of the bacteria. Once we plated them up and we were counting them, we would actually take the numbers that we used to identify our colony forming units. And we don't do a lot of math in microbiology. This is one math that we will be doing. I'm not going to quiz you on this yet until we actually get to that lab. But what we would do is we would take the counts, multiply it by our dilution factor, divide it by the amount that we plated, and then this would give us our colony forming unit, our number. These tend to be large numbers, so you notice that CFU is actually written in scientific notation. It's just a little easier to compare big numbers with scientific notation. If you've ever worked with a spreadsheet with a lot of zeros, it's really easy to misplace a zero. We certainly don't want to do that with doing our counts. That could be the difference between something being recalled from the market and something being safe or dangerous to use. So that's why to compare big numbers, we would use that scientific notation. More to come on actually doing that serial dilution, plating, and the calculation when we get to our CFU lab. There are also some other ways to count cells. When we are growing them in the media, of course, those cells are going to be living, but if we're interested in all of the cells, this particular test, the Petrov-Hauser chamber, allows us to do it on a microscopic level where we would actually take a sample, put it on a slide with a grid, and then we could count it underneath the microscope. That grid just makes it a little easier to keep track of our counting. And then we could take that and then we could calculate those numbers. So this allows us to capture both living and dead cells. We are not going to be doing this. We'll actually be counting from agar plates and not on slides. If we had very low numbers, this is sort of a sophisticated setup that we would use, the most probable number. This is more like an estimate of the number of bacteria. So we would do dilutions and then we would grow our samples in media. When that bacteria is growing and metabolizing, one of the byproducts of that bacterial metabolism is the production of gas, and that gas would come out as bubbles. So we could actually, over a period of time, count the number of bubbles, and then we could actually take that and then look it up in a chart, and that would give us an estimate of how much bacteria was actually in that culture. We are going to be counting fairly large numbers of bacteria, and this is a more sophisticated setup, so we actually won't be doing this method. But if you wanted an estimate of some low numbers of bacteria, this is a test that you could do. This is actually a test that I performed. Oops, I'm just going to go right back here. And this is using a machine that we actually use in chemistry. And this is a spectrophotometer. And what this does is this allows us to look at the turbidity and cloudiness and basically translate that into a numeric value. So the cloudier it was means that there's more growth, and that number on the readout of the spectrophotometer would actually be higher. So we would actually take our sample and then shine a light through it, and then we would get this number, this readout. So the bacteria that I studied for my thesis actually came from the desert in Israel. And what my advisor did was he actually washed they were kind of like pine needles. That was the type of tree that they came from. And he captured the bacteria. So my job for my thesis was to grow these bacteria at different conditions to see if they liked warm temperature or cold temperature, if they liked salt, what different types of growing conditions they liked. So I had to test a variety of conditions on my 11 organisms. And by using a spectrophotometer, it allowed me to test bacteria in a wide array of conditions so that I could actually get a readout. So if that well, I use these plastic trays with these little wells. They had like 100 wells in each of these trays. If I had a high readout, that meant that it was cloudy, it was turbid, and that meant that there was a lot of growth. That bacteria liked that condition. If the number was small and it was not very cloudy, that meant it didn't really like that condition. So it allowed me to quantify the growth and allowed me to tell which growing conditions that bacteria liked. So this is actually something that I've used in lab. There are lots of different factors that affect bacterial growth. Some would like a more neutral pH. Some might like it a little more acidic. Temperature, oxygen. Some need oxygen. Some would actually die in the presence of oxygen. Moisture, hydrostatic pressure. So could they withstand really high pressure? So if there were bacteria in the bottom of the ocean, there's a lot of pressure down at that level of depth. And so they would have to be able to withstand that. There's also some nutritional requirements as well. Some are going to require certain carbon sources and nitrogen sources, certain trace elements and vitamins. If some have really complex nutritional requirements and they're really difficult to grow, sometimes we term those as being fastidious, that they are really kind of difficult to grow in the lab. So these are things that we have to keep in mind when we're growing bacteria, because just like you have your favorite foods, they're going to have their favorite foods as well. Some of us like the room temperature cool, some of us like it warm. So bacteria, they're going to have those same likes and dislikes just like we do. Does that mean if we put them out of their optimal temperature range that they're not going to grow? No, it's just that they may not grow to their optimal. So we're going to be taking a look at some of these different physical and nutritional factors. So the first one is pH. So if you remember the pH table, pH basically means percent hydrogen or potential hydrogen. And this has to do with the hydrogen ion concentration and hydroxide ion concentration. At the lower end, so the lower numbers are going to be acidic. It's going to have a higher hydrogen ion concentration. The higher numbers are going to be more basic, and there's going to be a higher hydroxide ion concentration. So some bacteria will like a more acidic environment, so probably anywhere from like 1 to 5. And they're going to like those acidic conditions. Some are not going to be able to live at those acidic conditions. An example of this would be lactobacillus. So if you eat yogurt, if you look at the back of your yogurt container, it will actually have the live cultures that are in your yogurt container. One of those is usually lactobacillus. It likes a more acidic environment. Neutrophiles, they like a more neutral environment. Now, usually we think about neutral as being anywhere from 6 to 8 pH. So that's the type of condition that neutrophiles are going to enjoy. These happen to be the most pathogenic bacteria. Think about why that is. The reason for that is that we are mostly neutrophiles. So in most places in our body, especially like our blood and small intestine and large intestine, it's going to be more of a neutral environment. And so if the bacteria want to live in us, they're going to have to tolerate those conditions. There are some bacteria, believe it or not, that can actually live in acidic environments. Think about where it's really acidic in our body. Stomach acid is very acidic. There's not a lot that will actually live in that acidic environment of our stomach. We are going to talk about one bacteria that can, though. Bacteria that like it a little more basic or alkaline, these are going to be alkalophiles. They're going to like a more basic environment. So maybe on the scale of like 8 to 12. We talked about Vibrio cholera. Remember that shape that kind of looks like a comma or a little apostrophe? Vibrio cholera is more of an alkalophile. Now, the problem with growing alkalophiles in the lab, one of the things that I was mentioning earlier about metabolism is that as bacteria are growing and metabolizing, a lot of their metabolites are going to be acidic. So what happens with these alkalophiles, if we're trying to grow them in lab, is that because of their metabolic byproducts, they end up turning their environment acidic and they end up killing themselves off. So this is why when we're trying to grow bacteria in lab, our media has buffers in it. And if you remember, a buffer resists changes in pH. And so those buffers are going to help to either gobble up those hydrogen ions or spit them out, depending on which direction they need to go. And that's going to allow that pH to remain stable. And so our bacteria can grow in lab. So I mentioned that not a lot will actually grow in our stomach acid and the harsh conditions of our stomach. There is one bacteria that can actually tolerate that really low pH. And this bacteria is Helicobacter pylori, or H. pylori for short. And they believe that this bacteria is instrumental in causing ulcers. And so they started to do some research in about the 80s or 90s on ulcers. And if you've ever had an ulcer or known somebody with an ulcer, you end up with these lacerations in your stomach that can be very, very painful. And as they started to do some research, they found in the majority of cases of people that suffered from these peptic ulcers that there was a bacteria that was present, H. pylori. Now, I have heard stories about the doctor that discovered this, and people didn't really believe it. And so the story that I heard was that to prove his point, he actually drank a culture of H. pylori and gave himself ulcers. That supposedly is how the story goes. And lo and behold, they found that yes, H. pylori can cause ulcers. I wouldn't necessarily go so far as to say that all ulcers are caused by H. pylori, but they have found it in a majority of cases. My mother suffered from ulcers. In fact, she ended up in the hospital one time for a perforated ulcer. And so when they were doing this research and this information came out, her doctor actually tested her for H. pylori, and she was positive. So he put her on some antibiotics, and I think it was a pretty long regimen of antibiotics. It was a few weeks. I believe it was tetracycline, but I'm not entirely sure. And it was supposed to help to get rid of that bacteria. Her ulcers and the pain did lessen. She didn't completely buy into it, so she was still careful about some of those foods that she knew that were kind of triggering for her. It can be very tricky to treat because it can actually come back, but some people have actually had some good luck with the treatment. The reason that it can actually live in that stomach acid and how it can maintain its proper conditions is that it actually makes ammonia. So in its vicinity, it's almost like it's raining outside, but it's nice and sunny where this bacteria is, because it makes this ammonia, which raises a pH in its vicinity. And that's actually how it can live in that acidic environment. So temperature is a requirement that I think we all kind of have. We all kind of have our preferences. Some people like it really cold. Some people like it warm. Bacteria are no exception to that. So certain bacteria are going to like certain temperatures. So psychrophiles like it a little cooler. So they are happiest at temperatures of 15 degrees Celsius to 20 degrees Celsius. If you're not too familiar with Celsius, that would probably be about 59 to 68 degrees Fahrenheit. Mesophiles like it between 25 degrees Celsius and 40 degrees Celsius. So that's about 77 to 104 degrees Fahrenheit. We talked about how most pathogenic bacteria are neutrophiles. Most pathogenic bacteria are also mesophiles. Why? Because we're mesophiles. Our body temperature is 98.6 degrees Fahrenheit or 37 degrees Celsius. If they're going to live in us, they're going to have to tolerate that environment. Thermophiles, they like it hot. So they like about 50 to 60 degrees Celsius or 122 to 140 degrees Fahrenheit. So I mentioned my desert bacteria before. Now, thinking about in the desert is very hot during the day. It's also cool at night. So I tested the temperature that they preferred. And most people, when I asked them, what category do you think they would fall under? Most people would say, oh, thermophiles. Obviously, they live in the desert. It's going to be hotter. Guess what? They actually surprised us. They were psychrophiles. They actually did their best growing at cooler temperatures. Think about why that is. If it's hot in the summer and it cools off at night, and then you go out in the morning and you step on the grass, it's usually wet because of that dew. It has to do with that condensation. So my bacteria that I characterized, they were actually psychrophiles because at that cooler temperature, there was actually more water available. So that's when they were actually able to do their growing. So it was a little surprise to us, but then when we started thinking about it, like, yeah, there is going to be more water available at those cooler temperatures. So it does make sense that they are psychrophiles. When we're talking about the different conditions that bacteria like, oftentimes we'll use different terms. I mentioned fastidious before. That means that they're kind of picky. They're going to have special nutritional requirements. Some that have to have certain conditions would be obligate. They have to have that condition. So obligate aerobes, they have to have oxygen. Obligate anaerobes, they cannot be near oxygen. In fact, a little oxygen could actually kill them. Facultative means they're very flexible, so they can adapt to suit different conditions. Those are some different terms that as we're looking at the different conditions that bacteria grow in, those are the different types of terms that we will actually use. I just mentioned oxygen. Some bacteria have to have oxygen, others oxygen will kill it. We are actually going to be testing oxygen requirements of bacteria. We're going to be using a special broth. This broth is called thioglycolate. It has nutrients in it, but it also has an oxygen gradient. So based on where we see cloudiness, that turbidity, this can tell us the oxygen requirements of an organism. So there's more oxygen at the top and no oxygen at the bottom. So if we were to see cloudiness at the top, that means that the bacteria would need oxygen. We would call that an obligate aerobe, where it has to have oxygen. If it grows just a little bit below the surface, it likes a little bit of oxygen, but it likes a little bit of carbon dioxide as well. We would term that a microaerophile or a capnophile. Capnophile means that it likes carbon dioxide. Facultative anaerobes, they will grow in whatever condition. They are very flexible. If there's oxygen, they'll use it and they'll undergo aerobic respiration. No oxygen, fine with them. They will undergo anaerobic fermentation. Obligate anaerobes, they cannot be in the presence of oxygen. So if we were to grow an obligate anaerobe, on the bottom of our thiotubes, we would actually see that growth. We would see cloudiness. Once upon a time, we would try to grow an obligate anaerobe in lab, but it was so difficult to grow because if it got in contact with just a little bit of oxygen, it killed it. So we actually haven't been able to grow that particular organism in the lab for some time now. There are also some other physical factors, so moisture. So bacteria, they're going to need water just like we need water. We talked about those endospores. They are resistant to drying, so they can actually handle dry conditions because they will go into that endospore state, that dormant state. Hydrostatic pressure. Some bacteria can actually withstand really high pressure, like barophiles that we would see on the ocean floor. They can actually withstand that high pressure. Osmotic pressure has to do with tonicity and those dissolved substances in water. And we usually talk about osmotic pressure in regards to tonicity. So hypoosmotic or hyperosmotic. So halophiles, like my bacteria, could actually handle quite a bit of salt. And so they were termed halophiles. So some bacteria can actually withstand salt, some cannot. And it's a little easier, I think, to see from a picture. So this is a picture of these different conditions of osmosis and tonicity and our bacteria. Bacteria, they are going to be at the mercy of their environment, just like our cells. So if you look at the different conditions, so an isotonic or isoosmotic solution means that there's the same amount of solutes and water on both sides of the membrane. So inside the membrane and outside of the membrane. Cells are going to be their most comfortable at that isotonic condition. That's when cells reach that dynamic equilibrium. So we've got that equal amounts of solutes and water on both sides. If we were to put a cell, a bacterial cell, or even a eukaryotic cell in a hypotonic or hypoosmotic solution, that means that there is going to be more water outside of the cell. Water is going to move from an area of high concentration to low concentration. So since there's more water outside than inside, that water will actually move in and it would actually start to swell. And with our red blood cells, if we were to give somebody an IV with, let's say, water, those red blood cells would actually lyse. Because of the bacterial cell wall, if we were to put a bacteria in a hypotonic solution, we would actually have that water pressing against that cell wall, and we would actually get this turgidity. And so that cell wall is going to protect it a little bit from lysing, unlike our red blood cells. If we were to put a cell into a hypertonic or hyperosmotic solution, then there's going to be more water inside the cell than outside. Water is going to go from an area of high concentration to low concentration. Water will leave the cell and we would actually see plasmolysis. So we would actually see shrinking. Knowing this about cells, this tonicity and osmosis, this is something that we have used for years before we had things like refrigeration. So they would use brining, curing, and even high sugar content or high salt content to actually help to preserve our food. So if you put a bacteria in a really salty environment, plasmolysis is going to happen. If that water leaves the cell, bye-bye bacteria. It's not going to be able to live without that water. So in a high salt type of situation, so if we had foods that are really salty, there's not going to be a lot of bacteria that will actually tolerate that environment, and they're not going to be able to live. So this is something that we've used for food preservation for years and years and years, so long before we had refrigeration. This is also the reason why you might find me at the Olive Bar at Hannaford on a Friday night. I'm not a big fan of buffets, as I'm sure you can imagine, but there's lots of salt in olives, and so I feel a little better about getting olives from an Olive Bar, just because I know there's not a lot that can actually live in that salty environment. Because water is going to be really important to the growth of bacteria, there are going to be very few that can actually withstand those salt concentrations. Of course, our halophiles can do that, and not a lot of bacteria is going to be able to survive without water. We have done quite a bit of talking about endospore forming bacteria, and endospores are that protective feature. So when cells that can make endospores notice that conditions are declining, so not a lot of water, not a lot of nutrients, they will actually go into this dormant state. And we talked about during stationary phase, there are going to be some bacteria, if they're spore formers, they're going to start to go into that dormant state. So these endospores can withstand drying. They're also very resistant to cleaners as well. And if times start to improve, they can actually start to germinate and then go into that vegetative state. Remember, the vegetative cells, those are those metabolically active cells. So they could stay in soil for maybe years and years and years. And then if water comes back onto the scene, then they will germinate. So first, that spore coat would start to be broken down. It's going to start to take in water, and then it's going to start to grow. So it's going to start to go through DNA replication and making proteins, and then it would be back into that vegetative cell. So endospore forming bacteria are a big problem in not only clinical settings, but manufacturing settings as well, because they're really difficult to kill. Sometimes the best way is to actually kind of bring them back into the vegetative state, but that's not such a good idea. I am going to mention a couple of endospore forming bacteria. The first genus is Clostridium. And the first organism is Clostridium botulinum. And this is a spore-forming bacteria. Now, what is particularly scary about Clostridium botulinum is that it can actually cause botulism. And we usually think about botulism as being foodborne. So if you go to the store and you go to buy some canned goods and you see that that can is bulging, it's a good idea to stay away from it. Clostridium botulinum, this is an anaerobic bacteria. So of course they would love the anaerobic conditions of canned goods. So those are the types of things that you want to stay away from. Botulism can be very, very serious. It tends to be more of an intoxication. It has to be the toxins that's actually causing the illness. It comes on so suddenly within a matter of a couple of hours that it's not really enough time for that bacteria to be growing. It really has to do with the toxins that it's producing. The botulinum toxin is one of the most potent toxins known. In fact, an ounce can kill the entire population of the United States. That's how incredibly toxic it is. Of course, what do we do with this toxin? Hey, let's take this incredibly toxic substance and let's inject it into our face. And this is actually what we use. This toxin is what we use for Botox. Of course, it's very, very diluted. This is also one reason why if you are going to get Botox, make sure that you're going to somebody that's reputable and they're not doing their own dilutions. And what happens is it interferes with that muscle contraction, with that acetylcholine that's binding to the receptors, and it causes this flaccid paralysis. And so that's why we don't get that wrinkling, because we're not getting that muscle contraction. Another bacteria, which is an endospore former, is also in the Clostridium genus, and this is Clostridium tetani. And this is what can cause tetanus. Now, we can get our tetanus shot, which is good for 10 years. And if you get a cut, a lot of times they say if you step on a rusty nail, it doesn't necessarily need to be rusty, but there's the potential that there could actually be this bacteria and it could actually cause tetanus. And this tetanus causes this intense contraction. So what this toxin does is it interferes with the inhibitory actions that we need in muscle contraction. And so our muscles are constantly contracting. If you try to contract all of your muscles at once and try to hold it, it's exhausting. If you, let's say that you've been outside and you've cut yourself, you're like, I don't know, I think my tetanus shot is kind of recent. I guarantee you, if you Google pictures of tetanus, you'll be hightailing it to the doctor. It is just an intense, horrible disease. They also call this lockjaw as well, if you've heard of it. It can also happen and be spread through the raw stump of an umbilical cord with newborn babies. Now, you might think this is pretty rare. It is in this country, but in some parts of the world, this can actually happen. I had a student several years ago. She was actually a pediatrician in Brazil, and she said in the jungles of Brazil, this was actually very common. And they had many babies that they took care of that actually had tetanus. Many of them didn't make it. She told us the story about one child that was in the hospital for months and months, I want to even say years, that actually recovered. That was actually pretty rare. We mostly think about it like if we're gardening or if we're walking and we step on a rusty nail or something, but it can also happen in other ways as well. Now, if we want to grow bacteria, we need to have a suitable media in order for bacteria to grow. So the media that we use in lab and that they use in micro labs all over the world can be either selective or differential, or actually both. Selective means that it's going to allow some organisms to grow, but inhibit others. Differential means that it's going to allow different types of organisms to grow, but we can tell the differences between them. So we will actually be working with an agar this semester. It's called MacConkey agar. It's actually selective and differential. So it will allow gram-negative bacteria to grow, and it inhibits gram-positive bacteria so that gram-positive bacteria won't grow. So it's selective in that manner. It also contains lactose. There's also a pH indicator in the media. So it allows us to see the differences between cells. So we can see the differences between lactose fermenters and lactose non-fermenters. So the lactose fermenters, as they are metabolizing, those metabolites will actually change the pH, and we would actually see a color change. So in that sense, it's differential. A lot of times it's actually better to use a media that's both selective and differential because you kind of kill two birds with one stone. Some bacteria can be very fastidious, and we would actually have to enrich their media. So for example, Salmonella typhi, which causes typhoid fever, requires selenium. So if we were interested in growing that bacteria, we would actually have to add that to our media. So we will be working with different types of media in lab. Again, sometimes it's better to go with a selective and differential media just so that we can cover more bases. The rest of this chapter is sprinkling in some different bacteria and diseases, and I'm going to be talking about those in the second recording.